Welcome to the digital imaging track. We have some great talks lined up for you. I'm your moderator, Liron Pantanowitz from UPMC. We have two talks, then we'll have a break, another talk, and then later at the end of the day, this track will continue, but John Gilbertson will moderate the last talk. So the first speaker we have here is Jason Hipp. Jason is probably well known to several people. He did his fellowship at the University of Michigan last year in informatics. This year he's a junior investigator at the NIH. Jason has done some very groundbreaking work in automating laser microdissection, which is key today as our specimens get smaller and smaller and we're required to do more and more for precision diagnostics. To automate this process is a great boon both for clinical practice and for research, and Jason's going to share what he's done and his insights into the field. So, Jason, thank you.
Thank you for this opportunity to speak. My title is going to be Image-Guided Laser Microdissections. I'll be discussing clinical and research applications. So the objective of this work is to improve the laser dissection workflow by automating the cellular identification step. So we're going to redefine the involvement of the pathologist as a supervisory role. And it's also to develop an evidence-based understanding of the indications as to when and how to use laser dissection in the clinic to improve patient diagnostics and care. So here's a brief outline of my talk. I'm going to start with the background on LCM, talk about computer-aided laser dissection, discuss clinical implementation of laser dissections, talk about another technology called XMD, and then end with the future directions and conclusion. So background. So I'd like to start with an overview image. This is from the Applied Bioscience Arcturus website of just giving you a summary of what laser capture microdissection is. So you have your slide. You don't use a cover slip, and you put a cap on it because this is where the cells are going to adhere to. So as you see here, the cap is adhered to the tissue. And this is an overview look. And what happens is a pathologist annotates the cells that they're interested in. So you can see these tumor cells here are circled. And this is for a UV cut line in which the laser is going to come in and cut around it. And the green spots are for the actual infrared laser, which is going to melt the tissue to the cap. As you see here, this is going to adhere the tissue to the cap here. And then the laser is going to come around and cut it out. So the idea is after this is done, you can pull off the cap, and the cap has your cells of interest. So this has a UV cut system and an IR cut system, and there's different ways of using it. You can also have the UV cut line placed there, and then you could come in and manually add, or actually sometimes you could just use the UV cut line itself. And other systems just use the UV cut line and the tissue falls off the slide. You can place these manually or automated. Another option is just to use the infrared capture, in which you could just melt the areas of interest on the tissue to the cap, and you don't need to UV cut. So there's different advantages working with different slide types, and as you think into the clinical workflow, you're going to have to optimize these kinds of steps. So what happens is the cells are pulled off the cap's gone into an Eppendorf tube, and you can digest your RNA and DNA and protein. So just a brief history of laser dissection. This was developed at the NIH by the team here, and Dr. Leota and Dr. Buck. This is the first commercial, this was the first product that they made. It was the original NIH LCM machine developed in 1996, and it was published in Science. A year later, it was unique as it was followed by a commercial product in 1997. This is the Pixel. And currently, in 2007, this is the Arcturus XT, and this is the machine I did a lot of my studies on. So the current microdissection methods, there's currently 4 main platforms that people are using: the Zeiss, the Leica, the MMI, and the Arcturus. And they run about $250,000 each. So over the past 10 years, these 4 companies the industry has sold up to $750 million to $1 billion worth of cells combined. So what is the clinical research molecular diagnostics workflow presently? Well, you have a specimen, which can be from a patient or an animal model system. The tissue goes tissue processing, and you can see an H&E image of a prostate here, and this is your histopathology. This is then sent for molecular analysis, and then this information is fed back either for a diagnosis or for further research. So currently, with the molecular analysis, the tissue is either scraped or macrodissected. And the problem with working with tissue like that is that you can see in this tissue section, in the upper left, there's carcinoma. In the right, there's PIN. In the lower right, there's normal tissue. And then the left, we're showing here, there's also tumor-associated stroma. So this is normal, quote, normal cells, non-tumor cells next to the tumor cells. And there are molecular differences between this and normal stroma, and I'll show you that in the next slide. But then this doesn't even address the tumor heterogeneity, so even within the tumor, there's differences. So you see, if you just try to scrape or you cut these cells, you're going to be profiling a lot of different cell types. And this is what our group has shown, that the challenges of macrodissection, so even if you took a glass slide and you cut out the tumor cells, the stroma next to the tumor differs from the stroma outside. And this is shown here with microarray studies here. There's protein, EpCAM is shown to be different with the tumor-associated stroma. The endothelial cells next to the tumor are different too. So here's a question. When using non-dissected tissue, what will the molecular differences be between a sample that had few tumor cells and lots of tumor-associated stroma, or few tumor cells and normal stroma? So when you are identifying these differences, are they due to the stroma and not the tumor itself? So these are some things to think about for current molecular studies. So is there a research need for microdissection? There's been papers that's shown this, that using microdissection, you're able to identify differences in gene expression profiles. You can identify, it provides more information than just using the whole tissue. Other studies are showing that you can identify genes that are not otherwise found, and you could get additional information. So why isn't LCM more prominent in the prominent use clinically and in the research workflow? It's because it's very time consuming. So you can see this prostate gland, and normally what happens is that you have to fire a laser at each point to get the cells that you want. So a pathologist or a histopathologist has to sit there and fire those. Once you identify the cells, then you can pull those off the cap and you can see what's remained. So this rate limiting step is the operator time of the pathologist to identify and actually fire the laser for each of these cells. And you can imagine, you saw how long it took for these shots here. Current DNA studies use 3,000 to 10,000 shots. And proteomic studies use about 100,000 shots because you can't amplify the material. So since the rate-limiting step of laser dissection is the operator time of a pathologist required to identify the adequate number of cells, can you integrate image analysis algorithms to aid in the cellular identification step and reduce the need of a pathologist to a supervisory role? So I'd like to talk next about computer-aided laser dissection. So this project was begun when I was a resident at the NIH under Dr. Buck and as a fellow with Dr. Belize at Michigan. So the idea is you have a patient, their tissue gets processed into the histopathology, then an algorithm will come and identify those cells of interest. Then the laser capture microdissection tool will come in there and cut out those cells that were identified, and then those cells are sent for molecular analysis. So the software I was using for this is SIVQ. I'm just going to give a real brief background. It's a pattern recognition algorithm that uses ring vectors. The key features that it's a zero training workflow. It's interactive, it can run on laptops, and it runs at multi-length scales. And these are all key factors when you're integrated into the clinical workflow of LCM. And this code was written by Jerome and Yule. So how is it used? So an image is selected, it's searched, and a heat map is then painted onto the image. Here are some use cases. You can use it in the upper left to identify band cells, calcifications, fungi, and actually nuclear features with these are Reed-Sternberg cells. Another algorithm that I'm going to talk about how we're integrated into the workflow is PPMM. This was an algorithm developed by James Monaco and Anant Matabushi. So this algorithm is unique in that it identifies prostate cancerous regions based on luminal architecture, and it assesses the white space, not the tissue itself. So what we did is we've integrated SIVQ and PPMM into an algorithm cascade. And I'll show you this. So here's your ground truth tumor. So here's the ground truth, and here's the tumor. We ran PPMM on this tissue, and you can see that's in green, identified most of the tumor. Then we ran SIVQ, and this is a vector to recognize the epithelium, and that painted all the epithelium here. And then we did in a cascade approach where we ran SIVQ on the PPMM identified, and you can see how we're able to identify much more cleaner the tumor epithelium itself. Another tool that we use in the workflow is called image microarrays. This is a tool we recently published. It allows you to create a single digital slide containing the key diagnostic entities and appropriate controls, the benign mimics and the background features for algorithm screening and validation. And this is very important for the LCM workflow because you want to have your algorithm up and fine-tuned so that when you have your tissue specimens, you have frozen tissue, you got to work fast, so you have to have your algorithm fine-tuned. This is akin to TMAs for IHC screening. So you can use IMAs for algorithm screening. And also we developed a tool that allows you to core out the images just like you have a core tool to develop for our TMAs. Here's just a brief overview of the graphical user interface, is that you just navigate the slide, you capture the images, and these are preset windows from whole slide images, TIFFs, any kind of format. And what you can do is you can put them in this array maker, and then you can make collages. And this is what we did for one of the lung cancer studies, is that the tumor morphology was so unique that we were able to test our vectors for all these different morphologies, create heat maps, and then optimize the vector for the workflow. So again, here's the LCM SAVQ workflow. This is with the help of Jeff Hansen. So the important feature here is just you normally have a tissue block, you make a slide, you stain it, you image it, and we're adding these two steps. We do a digital enhancement, and we use SIVQ, then we import it back into the XT software. So one of the biggest challenges is of starting this project was improving the image quality. So it has to use tissue that is non-cover slipped, and you can see this is an H&E of non-cover slip tissue. So what we were trying to explore different methods to improve the index matching. We used ethanol, and it improved it a little bit. The other important thing is we needed stuff that would evaporate, because we don't want to affect the adhesion of the polymer to the tissue when you're firing it with a laser, because you can cause a lot of problems like that otherwise. So ethanol evaporated. We like that image quality. We then explored xylene, because xylene evaporates very quickly. So this is an H&E. We added xylene. Then we started doing image enhancements, in which we're able to improve the quality to here and then here. So then what we were able, now these are kinds of the images that we can actually use practically. So using xylene and this autocorrect function. And also we're exploring different stains. So we did an esophageal cancer study, in which this is the esophageal tumor, right here it's stained in blue. So we explored toluidine blue. And toluidine blue lights up the tumor very nicely, in which now this is very easy for an algorithm like SIVQ to come along and identify those. It also worked on the normal cells, because we had a lot of bleeding through when we were doing H&Es and stuff, but toluidine blue created a gradient, and we created a vector to recognize the gradient at the base, the basal cells here, and that was shown here. So we took a normal biopsy, ran a SIVQ, and pulled out those tumor cells, or the normal cells. We did the same thing for the tumor cells, and then we compared it to non-image guided or non-SIVQ LCM tissue. So these are our VQ. Those were the normal samples identified by VQ and normal, and tumor and tumor. And you can see how all the normals clustered with the normals and the tumors clustered with the tumors. So this shows that our workflow is not causing any changes in the biomolecules. We're also doing this, and this is one of my favorite images. This is of a dog prostate. And what I did is I found a unique morphology. This is a single nuclei with a prominent nucleoli. And then you can see a cleave and some clearing here. And you can imagine trying to count there's approximately 400 cells here I've counted. And trying to identify that feature in there could be very time consuming. We use SIVQ. I looked at where it found, and I confirmed it. And then I was able to pull out just those cells, and you can see the 11 nuclei and the corresponding cytoplasm as they projected into the lumens. And this is what remains. So this shows that you can find a specific morphological feature and pull out just those cells. Another thing what we're doing is integrating algorithms. So we used PPMM. So this is actually a prostate tissue section. So we use PPMM to identify where the tumor region is, and you can see that annotated in red here. And then when we went into the arcturs, circle that on the microscope, and you can see, so this is again the image, and this is when it's not enhanced. Circle the area. Then we ran our SIVQ epithelium vector on there. So we're able to pull out the epithelium, the tumor epithelium here. And this is what we pulled out. This is a low power view of the tissue. So next I want to talk about the clinical implementation of laser dissection, some of the obstacles, and how we're trying to integrate it into the cytology molecular workflow. So our group's goal is to develop an evidence-based understanding of the indications for the use of laser dissection in the clinic to improve patient diagnostics and care. So this study was done by Sanchita, Dr. Finlay, Dr. Raphael, Lee Chang, and Dr. Tangria, done at the NIH. So she did LCM to identify small numbers of tumor cells for EGFR and KRAS mutational testing from known lung cancers. So you see here in the bottom left, these are so she did this by hand, circled the cells of interest, and was able to dissect that, and did the KRAS and EGFR mutation. What she did is then she compared those dissected cells from those with previous known molecular status. She was able to detect the identical EGFR and KRAS mutations present in the patient's prior molecular assay in every case, and they were also able to consistently detect a mutation from as few as 50 tumor cells. And this is very important because often in cytology, you don't have enough tumor cells, or you get a few tumor cells that are scattered. So now, and those things are normally deferred. A patient has to undergo another biopsy, a potential another infection. They have to redo this, and hopefully they have enough. So this is another way of pretty much, it might be more time investment up front, but in the long term, it save a lot of time and money, and also help the patient much more. And so what the importance of this study is that by isolating more pure tumor cells, you can increase the sensitivity of mutation detection, and they're able to detect mutations of LCM-rich cases where the tumor was low and traditional methods of whole site scraping failed. So we were very excited by this result, and we asked the question, can SIVQ-LCM be integrated into this workflow for mutational analysis for cytologic lung adenocarcinoma samples? So the first thing to do, since the H&E images of pleural effusions of lung cancers are very similar to benign mesothelial cells, we use TTF1 to help us identify the cells. So this is a TTF1 stain. We did SIVQ to identify the tumor cells and dissect those out. We also did it on H&E. It's a much harder on the H&E, and it's more of an enrichment than getting pure tumor cells, but we showed that we can at least enrich for the tumor cells here, and we're able to dissect those out. So the next question was for us to address is, what are the, we had to identify and overcome roadblocks for the potential clinical implementation. And I've listed some of these here, and I'm going to give you representative, some images, some of the data showing how we overcame that. So again, how do we improve image quality even more? How do we have finer dissections to get pure single cells? How do we increase our laser spot placement? I'll show you why that's important. Increasing the retrieval yield, depending on whether you're using glass slides or membrane slides. Using various stains, because we know stains have an effect on the downstream biomolecule assays. We're also integrating post-dissection image analysis to help with cell counting. And then I'll show you how we're doing mutational testing on these samples after they've gone through this workflow. So one of the things is what we showed xylene and using the image enhancements, you can improve image quality, but sometimes for these kinds of assays, for especially lung cancers, that was we really needed high quality images. So what we thought of, we put a glass cover slip on the tissue, we imaged it, and that's here with this IHC. And then we did a SIVQ, we identified a heat map. Then we then took off the glass slide and dipped it up and down in xylene. The glass slide came off, we put it back in the machine, and because there's drift on the machine, and you can see the heat map drifted, and we noticed about 300 to 500 micron drifts consistently. However, we're able to actually just isolate this map and then just relocate that on top. So this is a little bit more time consuming, but when you need to do very important dissections, we use this method. And you can see here, so this is what an H&E non-stained image would look like, and this is with our method with glass. And we're able to use SIVQ to enrich for the tumor cells and dissect those out. Another way to improve our dissection efficiency was to increase and optimize our paint size, to have the UV laser cut and ablate the adjacent cells. So what we use SIVQ here to do is that we can easily identify these cells here, but we dilated whatever it identified, we dilated it to about 20 to 22 pixels, and you can see that they're a little bigger here. It's because when the laser cuts around there, it like burns the tissue around it. So we started to use this for advantage. So we went a little bit bigger than the normal cells, and when we cut just around the cells, we're able to kill the adjacent cells, so that thus we could get more pure cells when we dissected. And you can see here, we're getting just the cells. But you can also notice some of these spots didn't dissect out the cells. And you can see it's because there's no IR spot placed here. So that was the next thing we had to overcome, because when we're doing single cell assay, when we want to identify single cells for low cell count numbers, that was a very important thing. So we're starting to, we optimized the autoscan algorithm. This is the algorithm in which we place the IR lasers. This is built into the software. So using either the UV cut placement or the IR cut placement, you can see how we can optimize, we can easily change the number of laser spots we put. And we had to optimize this, especially for clinical workflow, because if we're identifying 200 cells and then the IR spot's not placed in there and it's not coming off, we had to understand why that was and how can we get around that. So then when we combine that, increasing the spot size, and then we do the UV cut, then the IR spot placements, and we use membrane slides. So what we do is we have it's a glass slide with a little membrane on top, and then when you're able to cut around, it pulls off the cells much easier because it doesn't have as much static attraction to the glass. And you can see, using this technique, you can see how we can consistently dissect pure single cells here. And that's the post-dissection. I'm just going to show you a few examples of using this technique, what we're able to do. This is a prostate tissue stain for an IHC for a basal marker. You can see the basal cell stained here. We enhanced the image. We ran SIVQ. You see how the heat map is placed down here where the stain is. We dilated that map, dilated the heat map, and this is what we pulled out, and this is what remained. We overlaid the heat map, and this is a good QA system we do. It's just to make sure whatever was painted got dissected out. We overlaid that on the tissue to make sure as a confirmation. And you can see here, this is where the basal cell layers are all taken out. And I think these are going to allow us to do some really cool molecular studies. We're also using various stains in this workflow. You can see up here is a fast red, the nuclear red here. We were able to do SIVQ, dissect out those cells. We use de novo red, another staining method. We did the same thing. And we're also using fluorescence now. So we're currently evaluating when do we use which method, what is the downstream effects, and we think that we're kind of going to describe a decision tree in which, depending on your downstream assay, depending on your case, we're going to be able to fine-tune each of these parameters. Another important thing where we realize, or another advantage with image analysis we can do, is that once we take out the cells, we could count those cells, and we can start estimating how much DNA we're going to have, thus determine how many molecular assays we can do. And this is going to be very important for clinical implementation. So this is an image dissected, taken right off the cap. So you, once you pull off the cells off the membrane slide, you look at the cap, you can image it, and you can see these are the stained nuclei. We then ran SIVQ to identify those nuclei, and we can fine-tune the vector threshold, and we with a manual count, and we can start using this to know how many cells we dissected, and I'll show you a little bit later on why that's important. And so we can determine, like, if you're doing a lung cancer and you want to do 10 molecular assays for 10 different genes, and you only have 20 cells, we know what genes, and we can tell that to the clinicians so they can highlight this and to know what they can do. So now we're also doing KRAS mutational testing using this workflow. So we took these pleural effusion blocks and we stained it with TTF1. We dissected out these 3 fields of view. You can see here this is a 6mm cap, and pulled out these 3 tissues. And with Li Cheng and Dr. Raufeld in the molecular diagnostics lab at the NCI, we were able to run and do assess for KRAS mutational testing. So just to step back at a high level, this is the pleural effusion lung adenocarcinoma workflow. What we imagine is that we have our stained tissue for TTF1. We do SIVQ. We can estimate the number of cells, 309 cells. We dilate the cells, dilate the heat map. We dissect out the cells. That's what remains. These are what we pulled out. We overlay the heat map to do a QA to make sure we got all the cells we want. Then we run SIVQ and get an estimate how many we have. Then we can determine what kind of assays we can do next. So next I'd like to jump and talk about a different dissection technology. This was developed by Dr. Tangria at the NCI. It's called expression microdissection. So this is different than the other platforms, such that you use IHC stained tissue, you place a film on top of that, and then you flash a light or a laser source, and whatever is stained adheres to the membrane, and you can just pull off the membrane, and the cells are attached there. And then you can do downstream analysis. And this gets away, this is much more operator independent, and you can do much more high throughput collection of cells. Jeff Hansen here at our group also adapted this technology to a commercial device, so that they used this, the same Arcturus XT here, and they were able to, again, same thing, so instead of flashing a light, they fired the IR laser throughout the whole tissue, and you can see here, here's your tissue, and here's what they're able to dissect, and that's what remained using this technology. And with Jeff right here, what we're doing here is we're doing a study in which we're comparing XMD and we're integrating SIVQ to aid in the XMD process. So here's your IHC stained tissue. Normally what you do is you would circle the area, and this would be your area that you're going to fire the laser, and it's very time consuming because you've got to fire a laser at each point, and you can see in this whole area here. And then you pull off the membrane, and these are the cells you get. However, now with, by integrating SIVQ in the workflow, this is going to target the laser specifically to where you want. So instead of firing the laser on the whole tissue, we use SIVQ to say fire the laser near here. And what we're able to do is increase our time tremendously. So if laser's just fired there, then we're able to dissect out those tissues. So I'd like to jump to some future directions and conclusion. So how do I see this computer-aided laser dissection implementation in the clinic? Well, here's your H&E image. I see there's research and clinical applications. So the research applications, I think you can use it for enrichment. So this is where you'd use an algorithm like SIVQ, identify the cells. Sure, it might not get every cell, or it might get some extra cells, but you could definitely enrich more, and then you could do your expression profiling assays. Or for clinical applications, I see it as more of a guide. So the pathologist doesn't have to annotate every cell. The algorithm will identify most of them. The pathologist will come in QA, add additional spots or delete spots. So you get very high purity. And these will be things for like KRAS mutation testing when you have very few cells. Again, the future, or how I also see the future of this workflow is that you have a patient. Their tissue undergoes processing for an H&E image. An algorithm will come along and identify the cells of interest. You're going to dissect it for molecular analysis. And then you're also going to get data from this molecular analysis, feed into precision medicine. However, by integrating this CAD analysis, which we're showing from the last couple of days' talks, we're going to get a lot of prognostic information. And then by adding that directly to the cells that you specifically identified, we think that it's going to be not just morphogenomics, but precision morphogenomics, because these cells have been profiled specifically for, and we know the exact molecular phenotype of these morphological phenotype characterized cells too. So another thing is we can think of is that you can see with these devices, they all have microscopes. We can also think of integrating a whole slide imaging scanner to improve the image quality. And we're also going to be continuing to work towards integrating various algorithms and technologies into the LCM XMD workflow. And we're going to try to take advantage of each strengths, and we're very interested in collaborating with different algorithms, such that we can use either SAVQ LCM, SAVQ XMD, PPMM SAVQ, so you can see how we're going to try to leverage each algorithm's advantage with each dissection technology advantage. So again, the future directions of our work is the evaluation of if and when to incorporate laser dissection into the clinical workflow. We want to establish and assess the use of reliable, reproducible, high-throughput methods to address the clinical need. Sometimes you won't need it, sometimes you will, and that's what we want to try to identify. We're also interested in getting CLIA certification for the use of this into the clinical workflow. We also want to improve the speed and precision, and so that we can do downstream large amounts of tissue for proteomics and next-gen sequencing experiments. So here's the current workflow when you have a patient. They have their tissue. Pathologist reads, makes a diagnosis, and you have your molecular diagnostics also. And that's the current precision medicine. I think that computer-aided laser dissection will be a bridge between these two, such that you'll have not only the diagnosis, but you'll have the CAD analysis and the image analysis results. You also have the molecular diagnostics for this. And I think this is going to be the true precision morphogenomics. So in conclusion, we're developing an evidence-based understanding of the indications for clinical use of laser dissection to improve patient diagnosis and care. We have developed an automated image-guided laser dissection workflow pipeline. We have demonstrated proof of principle that this workflow can be integrated into the cytomolecular clinical workflow for lung cancer pleural effusions. And we believe that CALD, this is a computer-aided laser dissection, holds promise to bridge morphology with molecular diagnostics while maintaining a high-throughput automated workflow. And I'd like to give a special thanks to our collaborators and a special thanks to Dr. Belize and to Dr. Buck. Thank you.
Jason, thank you. That was a fantastic talk, and it's impressive how much you've accomplished in such a short time frame. So I'd like to open it up to the audience for any questions. If you have for Jason, please feel free to ask.
Excuse me. Let me cough into the microphone. Lovely stuff. I wonder how you think a little bit more about the whole slide integration with the dissection. Do you see an actually integrated instrument or moving the slide from one to the other, and how would you address the image quality issues that you've outlined?
Yeah, that's a very good question. That's what we're just exploring that right now. I think I don't have a specific methodology right now, but I think that, because we also have non-converse slip tissue, and then if we put xylene on there, it's going to ruin the scanner. So whether we use an actual whole slide imaging scanner or the components of such a scanner like that built into a microscope, I think that might be a little easier right now, and then just have the things fed in directly.
Jason, with more and more studies showing the heterogeneity of mutations within tumors from cell to cell, how do you see this technique applying to looking at that issue?
Yeah, good question. I think that's going to be addressed with our CAD and image analysis algorithms as we're identifying, I think there's some of the talks from the HEMA lecture were showing how you can subclassify these different tumors and where they're located and stuff. And I think once you have your algorithm identifying that, which they're doing, I think the next thing to do is not just stop at the CAD analysis, but also follow directly to the molecular analysis and compare that in general to those cells versus the whole tissue scrapes in general to see where you're coming from and for confirmation.
How much time do you save with SIVQ to collect a correct population of N cells compared to traditional LCM?
Yeah, that's a good question. So the thing is with that is it depends on the tissue and what kind of cancer cells we're going after. So for IHC stained tissue, it's very easy. The algorithm runs fast. It's more on the image processing right now. And the second thing is that it's a little more time consuming now because we're just using commercially available machines and we have to export and stuff, so we can't fully integrate. So that's the one thing we want to do, is start integrating all these technologies so we can kind of customize and design our own platform for this. But mainly it's the image processing tasks. Like for the pleural effusions, the H&E image is very hard to identify, and getting different vectors, and that's time consuming. However, but it also alleviates the need of having the pathologists so that those can be confirmed once the vectors are established.
So Jason, I have a question for you, which is, at the moment there's a lot of research and investigation going on, and where in the pipeline are you in making this commercially available so that labs can purchase a device to perform this computer-assisted laser dissection for them?
Yeah, so right now we're getting ready to publish the workflow and a lot of the things that we overaddress and how to do that with the XT. And right now, an SIVQ is open for academic collaborations. So we see if you can contact Dr. Belize for the algorithm, we can definitely help you get your clinical workflow. Because what we want to see is we want to see this pushed into the clinic with as many groups and confirmed. So if anybody is interested in this, we'd love to help them get their core facilities set up such that these things, because I mean, I didn't address this, but we've been talking is that there's so many areas that we want to address for the clinical implementation that this just can't be done by one group. We're going to really need a diverse group, different algorithms. We don't know which technologies, and actually identifying the use cases when it is and when it isn't, and exploring these different technologies.